Dear students, Assalamu alaikum and welcome to office.com.pk. Dear students, in the last lecture we have discussed about Watson and Crick model of DNA, which they have presented in order to explain the structure of DNA molecule, how it is arranged and how it is properly organized in order to store the genetic information. Today we are going to move forward and we are going to study the DNA replication, how the process of copying the genetic information continues from generation to generation. And for this purpose, we are going to use Watson and Crick model for our basis. So, when we study Watson and Crick model, it suggests that the replication of genetic information is going to be dependent upon complementarity. Complementarity. Now this is a very important terminology which you have to keep in your mind while studying the DNA replication. And on the basis of complementarity, very much hypotheses were presented in order to explain how DNA replication is carried out from generation to generation. Now, complementarity suggests that if we unzip a molecule of DNA, then in order to unzip a molecule of DNA, we need only to assemble the appropriate complementary nucleotides on the exposed single strand to form the two daughter complexes with the same sequences. For example, if this is a DNA molecule and you have to unzip this molecule, then what will you do? You need to provide a complementary strand for each strand. Now the two strands will be separated, but they will be stabilized by two different strands, and this will result in the formation of the daughter DNA strands. These strands which are newly formed, they will have exactly the same sequence of the strand that is lost by each parent strand. Now this kind of replication is known as semi-conservative. Semi-conservative. Why is it called so? Conservative means reserving the actual form, reserving the actual copy. So semi-conservative means that one strand is kept the same or it is conserved, and semi word is used because half is conserved and half is lost, and that conservative means the sequence is conserved. At the end we'll have the two exact copies of the parent DNA, so the sequence will be conserved, but the strands, they are not conserved, they'll be separated. In semi-conservative replication, the strands of the duplex separate out. Each will act as a model or mold along which the new strands will be formed, and the nucleotides will arrange themselves according to the sequence of the parent strands. In this process, by separation of two strands, the primary structure which was actually present is not has been conserved, whereas the secondary structure is disrupted. Now the other hypothesis, this was the first one, that the DNA replication which is based upon Watson and Crick model, it is semi-conservative. Now we will discuss other hypotheses that are presented in order to explain the DNA replication. First one was the conservative model. First is semi-conservative, the second is conservative. It will say that the whole DNA molecule, whole parent DNA molecule will be conserved, and the copies are made according to this one. For example, you take a page and you make a photocopy of that exactly alike, but you keep the original page as it is. So that is the concept of conservative model. You'll have the whole parent DNA conserved, and the new DNA is formed according to the sequences of the parent DNA. It will not require the separation of the strands of the parent DNA. Now, another or third one hypothesis was the dispersive model. First is semi-conservative, the second one is conservative, and the third one is dispersive. Dispersive model predicted that the parental DNA will become completely dispersed, and each strand of all the daughter molecules would be a mixture of old and new DNA. So it will state that DNA molecule is disrupted, completely disrupted into nucleotides, not the strands. Semi-conservative says that the two strands are formed. Conservative says that no strands will be separated. The new copy will be formed exactly like the parent DNA molecule. And dispersive model states that the DNA molecule will be disrupted into different nucleotides, and in the formation of new DNA molecule, these nucleotides will rearrange themselves, and the new molecules which are formed, they will be the mixtures of the existing nucleotides. So they'll have a mixture of the new nucleotides as well as the old nucleotides. So this is the basic concept about the hypotheses that have been presented for explaining the process of DNA replication. First was semi-conservative, second one is conservative, and the third one is dispersive model. Semi-conservative states that half of the strand is conserved and half is donated to the daughter nuclei or daughter DNA, whereas conservative states that whole parent DNA molecule is conserved and the new one is formed, which will be exactly the copy of the parent molecule. The dispersive model states that the DNA is completely broken or disrupted into separate nucleotides, which will rearrange and become a mixture of old and new nucleotides in the daughter DNA molecules. Now we will see the experimental verification, which will explain which of the hypotheses is correct in explaining the replication of DNA.